Welcome everyone. In today's lecture, we'll try to complete another chapter which is from 12th NCERT, chapter number 10. It is Microbes in Human Welfare. So again, this is one short lecture. In one lecture, we'll try to complete the whole chapter, chapter number 10, 12th NCERT, Microbes in Human Welfare. So the introduction starts the chapter in this manner, like besides macroscopic plants and animals. Macroscopic organisms means which are visible for us, they are plants and animals. Besides macroscopic plants and animals, microorganisms are the major components, right? Microorganisms are the major components of the biological system present on the earth. So that means microscopic forms are there, macroscopic forms are there. Under macroscopic, plants and animals will come. Under microscopic forms, microorganisms will come. They'll make the biological system on the earth. And then they are asking, like, how many kingdoms are there? R.H. Whittaker has classified 5 kingdoms. Out of these 5 kingdoms, under how many kingdoms are we going to keep microorganisms? The author has asked the question. What is the first kingdom? Monera, where bacteria are there. So this is a microorganism kingdom only. The second kingdom is Protista, which also includes unicellular microscopic forms only. The third kingdom is fungi. Fungi are also microorganisms only. Then plantae and animals, they are macroscopic. So the author has asked how many kingdoms contains microorganisms. First kingdom, second kingdom, third kingdom. Monera, Protista and fungi contain microorganisms. Plantae and animae doesn't include microorganisms. Now next question the author has asked, which are microscopic? Means on the bread we see rhizopus, fungi, we can see it, right? So that means it is not microscopic. Whereas the first kingdom, bacteria, we can't see with naked eye, we can see only under microscope. Now protistants like euglena, amoeba, all those things come under protistants, that we cannot see under naked eye, we can see only under microscope. See means how many are microscopic, means the first kingdom and second kingdom are microscopic. How many kingdoms contain microorganisms, means 3 kingdoms contain microorganisms. And talking about the introduction to microorganisms, we can tell microorganisms are omnipresent. What do you mean by omnipresent? They are present everywhere, in air, water, soil, everywhere they are present, and they show diverse habitats. What do you mean by diverse habitats? They are present in such an areas where no other life forms will exist. They are present in such an areas where no other life forms will exist. Like, can we give some examples where no other forms of life can exist? If we take geysers, where we use hot water, geysers where the temperature is still 100 degrees centigrade, there no other life can be seen but microorganisms are seen there. They are thermophiles. Bacteria which can survive at high temperatures are called thermophiles. Now deep inside the soil where oxygen is not there, there also microorganisms are seen. Then what are such type of organisms called? Anaerobic organisms. Now under the thick layers of the snow, under such chilled frosting conditions also microorganisms are there. What are they called? Psychrophiles, right? Then they are seen in extreme acidic conditions also, they can survive. What are they called? Acidophiles. That's what we told, like microorganisms are having diverse habitats where all other life forms are not existent, their microorganisms are there. Means at 100 degrees centigrade thermophiles are there. In deep inside the soil where oxygen is not there, anaerobic organisms are there. In the thick layers of frozen snow, psychrophiles are there. In acidic environment, acidophiles are there. Understood, children? Then microorganisms are diverse. How many types of microorganisms are there? In NCERT they have given protozoans, malarial parasite is a protozoan, amoeba, euglena, paramecium. So they are microscopic only. So microorganisms are diverse. They include protozoans, they include bacteria, fungi and viruses also, right? Now viruses, normal microorganisms you can see it under light microscope, but viruses are so tiny that we can see only under electron microscope. That means they are also microorganisms, right? So protozoans, bacteria, fungi, viruses, let it be a plant virus or animal virus, and there are some deviations from virus. If you take a virus, virus has a protein coat outside, which is called as capsid, and it has its genetic material inside. Genetic material inside surrounded by a protein coat together is called as virus. But there are certain forms where only genetic material is there, naked genetic material, RNA is there. Then what is it called? Viroid. Only RNA is there, naked it is, not covered by a protein coat. What is that called? Viroid. On the other end, if you see, highly coiled protein only will be there, genetic material is not there. Highly coiled protein is there, genetic material is not there. Then what is that called? Prion. Infectious proteinaceous particles are prions. Infectious naked RNA particles are called viroids. Together, viruses. All these are microorganisms, right? And for a normal person, if you tell microorganisms, immediately they'll tell, yeah, they cause diseases, right? So don't think that all microorganisms are harmful, they are disease causing. There are many microorganisms which are beneficial, which are used for us in day-to-day lives. That is what our chapter about microorganisms in human welfare. We're going to see this chapter like what are the beneficial activities of microorganisms, right? We know microorganisms, bacteria, fungi, viruses and all, they cause so many diseases. But now let us see the other end. Let us look at the beneficial activities of microorganisms, right? So that is our chapter. Now in this chapter number 10, if you see the content, if you see the map of the chapter, they have given 6 topics. Microorganisms in household activities, microorganisms in producing industrial products, then microorganisms in sewage treatment, microorganisms in biogas production, microorganisms which are used to kill pests and insects, right? And microorganisms as fertilizers. These are the 6 topics which we are going to cover through this chapter. Now before going to the first topic, microorganisms in household, now there are some pictures what they have given. I draw the pictures on the board, so when we finish off this, then the board will become free, then we can start with our topic microorganisms, useful microorganisms in producing household products. Now in NCERT textbook, they have given these pictures, children. I just took it on the board. Now here these 3 are bacteria, these 3 are viruses, and these are the culture plates where we are trying to grow bacteria and fungi. Let us see the diagrams also. See, NTA examination or NEET examination is about perfection. Now diagrams you should be perfect, labelings you should be perfect, NCERT content you should be perfect, right? So that's why don't ignore the diagrams also, don't ignore the labels also. Now if you see this diagram, I told these are bacteria. What is the shape you can see here? They are rod shaped bacteria. Rod shaped bacteria, I told bacteria is not visible with naked eye, you have to see it only under microscope. How many times they have magnified it? X denotes magnifying power. Under microscope, when you magnify it 1500 times, so this much shape we can see it. Rod shaped bacteria is called as bacillus. Bacillus which has magnified 1500 times, this much will be the size of the bacteria. Now next one is a round shaped bacteria, spherical bacteria. Spherical bacteria are called as cocci, which are also magnified by the microscope, same 1500x, 1500 magnification. Now next one, they have taken only one particular bacilli and they have magnified it to 50,000 times. X denotes magnifying power. So when you magnify it to 50,000 times, then you can see clearly one bacteria. We can see the topological view also. We can see the flagella also, the locomotory structures. So when we magnify it 50,000 times, then we can see the topological structure, right? So these 3 are the bacterial images. Now coming to the viral images, they have given 3 viruses. The viruses which infect bacteria are called as bacteriophages. The viruses which infect bacteria are called as bacteriophages. Now the typical shape of a bacteriophage is a tadpole shape. The typical shape of a bacteriophage is tadpole shape. It has head, this is the collar, this is the body, and this is a basal plate, and these are basal pins, hexagonal basal plate. At each corner it will have a pin like structure, and these are tail fibers which are called as prongs. What are the parts? I told you not to ignore the labelings also. The virus which infects bacteria is called as bacteriophage, and the parts are, it has an icosahedral head in which the genetic material will be there. It has a narrow collar, this is the body sheath, and then this is a hexagonal basal plate. At the corners they have basal pins and they have tail fibers. Tadpole shaped bacteriophage structure. It infects bacteria, it will kill the bacteria. Now this is an adenovirus. Adenovirus, it's an animal virus. This is a bacterial virus. This is adenovirus, right? It is having only head like structure, it's not having the body and tail fibers and all. This adenovirus, a virus means poison, means viruses are not beneficial, viruses are dangerous only. Now this adenovirus, what does it cause? It causes respiratory disorders. Adenovirus which causes respiratory disorders. Now we have seen bacterial virus, we have seen animal virus. Now let us see a plant virus. Now the plant virus which causes mosaic disease in tobacco is called as TMV. Now all the research activities, initial research activities were conducted in this virus only, TMV, tobacco mosaic virus, which comes in your 11th standard. So tobacco mosaic virus, compact rod shaped structure it is. Now this is one TMV, another TMV, another TMV. All these are tobacco mosaic viruses which have been observed under electron microscope. How many times you have magnified so that you are able to see this structure? Means 1 to 1.5 lakh times they have magnified. 1 to 1.5 lakh times they have magnified. Then we are able to see the structure of a virus in this manner. So plant virus, animal virus, and bacterial virus. Now we can culture bacteria and fungi in the labs. Now we can prepare a nutrient medium. That nutrient medium can be poured in petri dishes, and you can add water, you can add soil, because microorganisms are present everywhere. And then when you keep it under optimal conditions, there is an instrument called incubator. So prepare a petri dish, add the nutrient medium to it, add the soil sample or water sample or buttermilk where bacteria, microorganisms are there, and then keep it inside a chamber which is called incubator where optimal conditions will be maintained. Then within 24 hours or 48 hours bacteria will grow. Now like this the bacteria will be grown, we can see here. These are called as colonies of bacteria. Don't think that each one is one bacteria, it is a colony. It is a colony. You cannot see bacteria with naked eye, right? So these are colonies. Each colony can contain hundreds to thousands of bacteria. These are the bacterial colonies which have been grown in a petri dish. Now if we want to grow the fungal colonies, same protocol. We have to take a nutrient medium where fungi can grow, then take it in a petri dish and add the fungal sample. Now these are the fungal colonies growing in a petri dish. Children, these are the pictures which they have given in NCERT before starting the chapter. We understood. Take a screenshot, then we will start with the first topic, microorganisms in household products. Take a screenshot of this. Right, so can I erase this? Now let us discuss about microorganisms in household products. Means what are the microorganisms and what do they prepare the products which are we going to use in our normal household activities. Now under that so many examples are there. We will list one by one and we will see. This is an easy chapter, so then it should be a scoring chapter for you, right? It is microorganisms in household products. Microbes in household products, right? This is our first topic, 10.1, right? So microorganisms in household products. Under that, the first example what we can tell is curd making. Milk getting converted to curd is a microbial action only. The first one is curd production, we can write. Now, how do we make a curd? You should ask your mother, right? So, you can take milk, boil the milk. So boiling will give you the temperature which is required. Now the milk contains a bacteria. What is the bacteria which is present in the milk? It is called Lactobacilli. Bacilli means rod-shaped bacteria. Milk contains a bacteria called Lactobacilli. This Lactobacilli and other lactic acid bacteria, only one type of bacteria is there? No. Lactobacilli is also there, and other LAB we will tell. LAB stands for lactic acid bacteria and other lactic acid bacteria which are present in the milk. What do they do? So they convert the milk sugar. What is the milk sugar? Milk sugar is lactose. The milk sugar is lactose. They convert the lactose, they break the lactose into lactic acid. They break the lactose, the milk sugar, into lactic acid. Now it has created an acidic condition. The bacteria will do this. Now this lactic acid, what it will do? Acidic conditions. The milk contains a protein. The name of the protein is casein. The milk sugar is lactose. Milk protein is called casein. Casein in acidic environment. Casein in acidic environment. Casein was soluble. Now what happens in acidic environment? It becomes insoluble and it starts precipitating out, means liquid is trying to become solid now, and it combines with calcium and it becomes calcium para caseinate. Now it is soluble, so it is liquid. Now it is insoluble, it will become solid. It is insoluble, so it will become solid. So if it is liquid, we call it as a milk. If it is solid, we call it as curd. Since it contains sugar, milk will be sweet in taste. Since it contains acid, curd will be sour in taste. So this is a mechanism, and this process of conversion of lactose to lactic acid is called fermentation. This is called fermentation, and we tell that curd is more nutritious than milk. So curd, we tell it is more nutritious than milk. Why is curd more nutritious than milk? Means the bacteria which are present in the curd, what do? They synthesize more and more vitamin B12. So vitamin B12 very little will be there in the milk, whereas curd contains more quantities of, it contains more quantities of which vitamin? Vitamin B12. And why are we concerned about vitamin B12? Vitamin B12 helps in hemopoiesis. It helps in blood making, and blood is a transporter, right? So blood is required for the functioning of all the organs. Vitamin B12 is present in curd. Very little is present in milk. So that means curd is beneficial, certainly, when compared to milk. And this bacteria which are present in thousands and thousands in the buttermilk and in the curd, now they go and they stay in our intestine, stomach and intestine, and they check the growth of unwanted bacteria. Now if you are attending my class here, at this time if any match is going on or some unused other activity is going on, so you will not attend that, right? Means your time is not getting wasted. In the same manner, in our intestine, if there is space, unwanted pathogenic organisms will grow. Now this bacteria, what it will do? It grows in our intestine and it acts as a probiotic. It will not allow the unwanted pathogenic organisms to grow. So that means the lactobacilli acts as probiotic, or the curd acts as probiotic by checking the growth of unwanted microorganisms in the alimentary canal, in the intestine. So all this is information about curd. Now what do? We boil the milk and we will add a little curd sample which acts as an inoculum. We add little curd sample which acts as inoculum. This inoculum, the curd sample, it contains thousands of bacteria. It contains thousands of bacteria. Now they start the process very fast, right? So this process is called fermentation. This is the first product under household. When we have to discuss about the second one, let us talk about idli and dosa. Let's talk about idli and dosa. In idli and dosa, we use rice and we use black gram dal. We soak it and we grind it. We grind it and we store, we ferment it overnight, right? So when we are doing fermentation, now rice is also dried material, and this one black gram is also dried material. When you are soaking it, when you are hydrating it, and when you are grinding it, when you are salting it, and when you are closing it, maintaining anaerobic conditions, the microorganisms which are present in the air, like Streptococcus faecalis and all, the microorganisms which are present in the air, they come into that dough and they start fermentation when you maintain that anaerobic conditions. And what do they do for the dough? They give that puffed appearance for the dough. When you grind the idli batter, and if you make idli immediately, it will be very hard, but try to make it next day after 6 hours or 8 hours, fermentation will be done. So then it will be fluffy, it will be soft, and that fluffiness is because of the carbon dioxide. Carbon dioxide will come when fermentation happens. So in idli and dosa, so what happens? Bacteria will be there, and the bacteria, they ferment the dough. Right, the bacteria, they ferment the dough, and doing fermentation they release carbon dioxide. This carbon dioxide is responsible for the puffed appearance. The carbon dioxide is responsible for the puffed appearance of the softness of the idli and dosa. This is the second household product. And then when we have to talk about the third one, we can talk about bread. Bread making. Again, here also we use wheat flour, which is called as dough. Now in this dough we add an yeast. Yeast is used in bread making. The name of the yeast is called Saccharomyces cerevisiae. What's the name of the yeast? It is called Saccharomyces is the genus name. Cerevisiae is the species name. Saccharomyces cerevisiae is a yeast which is used in bread making. What it will do? The same thing, it will do fermentation. So the dough contains starch. It breaks the starch into glucose, it breaks the glucose into ethanol, alcohol, and carbon dioxide. So in bread also, that porosity, that fluffiness is because of the carbon dioxide only. We like the cake, sponge cake, because of its softness. And who is doing it? Yeast is doing it. What's the name of the yeast? Saccharomyces cerevisiae. That is why yeast is also called baker's yeast. Saccharomyces cerevisiae is called as baker's yeast. So we discussed three products here. First one is curd production, second one is idli and dosa. So dokla is also there. In dokla, instead of black gram dal, we use green gram dal, right? We use green gram dal. That's the difference. Here we use black gram dal, here we use green gram dal. But the process of fermentation, that puffiness, carbon dioxide production, fermentation is the same. The third one is the bread making, where we use yeast. Take a screenshot. We will continue with the remaining. Now, fourth one, what we have to discuss under microorganisms for household products. So see, if you have a notion that microorganisms, all microorganisms are pathogenic, disease causing, then remove that from your mind, because microorganisms are producing many products and we are eating them. We are eating them. Now the fourth one, we can talk about toddy. Now, in southern part of India, there are palm trees. There is a particular palm tree which is called as Caryota uranus. Now what is this? This is a palm tree. Now to the palm tree, what they will do? So they will climb up the tree. They'll climb up the tree, they will cut the phloem through which the sucrose is passing, and they'll damage the phloem, and they'll keep a pot there and they'll tie the pot there. So that means the sap, the sucrose sap is getting collected in the pot. Air contains microorganisms, and here is the food, sucrose is there. They start fermenting. By the end of the day, by evening, what happens? A crude alcohol will be produced. Means the sugar is getting converted to alcohol by the microorganisms which are present in the air. So that traditional drink is called as toddy. Okay, so it is prepared from a plant, palm tree called Caryota uranus. So the fermented sap is consumed as a drink, which is called as toddy. This is the fifth product we can take. Next we can talk about fermented fish. Fermented soya sauce, we use it in Chinese preparations, and in Kerala fermented bamboo shoots. Inside that hollow fermented bamboo shoots, they prepare many delicious dishes. So fermented fish, fermented soya and fermented bamboo are also edible only, right? Fermented fish is one example. Then fermented soya bean, and next one is fermented bamboo. All these are the examples. Now, the oldest product of microorganisms, where we use microorganisms, is cheese. All of us are aware of cheese, right? Now, there are different varieties of cheese, and microorganisms are used in the production of all those different varieties of cheese. Now, cheese has a particular type of cheese, has a particular texture, particular aroma and all. Bacteria and fungi are used to give that texture, and they are used to give that aroma. Now, for example, if we take a cheese called Swiss cheese, right? Now, Swiss cheese is also called royal cheese. Swiss cheese is also called royal cheese. Now, this royal cheese, what is the characteristic feature of the royal cheese? Means it has big holes on it. This cheese has big holes on it. Now, these big holes are nothing but the carbon dioxide bubbles. So, big holes because of the carbon dioxide gas which got trapped. And this Swiss cheese, royal cheese, right, is prepared by the action of a bacteria called as Propionibacterium shermani. So in the production of Swiss cheese, which bacteria is used? The name of the bacteria is Propionibacterium. The species name is Shermani. Propionibacterium shermani is a bacteria which is used in the production of Swiss cheese or royal cheese. There are fungal cheeses also. When it comes to the fungal cheese, we'll take down the names. One is called as Roquefort cheese. The other one is called as Camembert cheese. Now, since I told these are fungal origins, what are the fungi which are involved in producing this Roquefort and Camembert cheese? These are flavored cheeses. Now, Penicillium roqueforti is an organism fungi which is used in Roquefort cheese. Penicillium camemberti is used in production of Camembert cheese. So, the fungi is Penicillium roqueforti is the fungi which is used here, and Penicillium camemberti is the organism which is used in preparing Camembert cheese. All this, how many we listed? We listed 8 under microorganisms under household. Take a screenshot, then we will go to the second topic. What is that? Microorganisms in industrial products. Okay, so we finish this. Take a screenshot. Right, now when it comes to the next topic, what is that? 10.2 microorganisms in industrial products. There are many industrial products, right, where microorganisms are used, but we cannot do all of them. The main industrial products are alcohol, beverage industry and antibiotic industry, pharmaceutical industry, that we will discuss. 10.2, our topic is microorganisms in industrial products. Microorganisms in industrial product is our next topic. As I told you, many microorganisms are used to produce many different types of industrial products, but the main two things which we are going to discuss here under this topic are, the first one is beverage, alcoholic beverage production, and the second one is antibiotic production in pharmaceutical industry. So we are going to talk about beverage production and antibiotic production. Now, when we are talking about industrial scale, so when we have to produce it in large scale, which we call it as industrial scale, now you need big containers, such a big containers. Those containers are called as fermenters. Where do we do this process of alcohol making and antibiotic making? And a big vessels which are called as fermenters. In NCERT, they have given a big steel container, a fermenter in which the fermentation is being carried out. Understood? Now, let us talk about beverage industry first. alcohol industry. Right? So again, which microorganism is used in producing alcohol? Ethanol means it goes with yeast only. The name of the yeast is Saccharomyces cerevisiae. Have you heard Saccharomyces cerevisiae? Yeah, right, in bread making. Means Saccharomyces cerevisiae is called as baker's yeast because it is used in bread making. It is also called as brewer's yeast because it is used in alcohol making also. So the yeast which is called as Saccharomyces cerevisiae, which is used in brewage industry, so it is called as brewer's yeast. It is called as brewer's yeast. Now what it will do? So it will convert the sugars which are present in the raw material into alcohol and carbon dioxide by producing an enzyme called zymase. Now depending upon the raw material, how many type of alcoholic beverages are there? Now depending on the type of raw material, we can divide the alcoholic beverages into different types. Now if the raw material is barley, if we take barley juice, then beer is obtained. If the raw material is fermented rye, then gin is obtained. Fermented rice, sake is obtained. Like that, depending upon the raw materials. Let us see. If the fermented material is barley, then what do we get? I told we get, if we use fermented barley, then we get beer. Yeah, B with B. Now if we use fermented rye, then what do we get? Fermented rye gives us gin. And next, fermented rice. From fermented rice, we get an alcoholic beverage called sake. Now, fermented potato. From fermented potato, vodka. And from fruit juice, right, wine and brandy. From fruit juice, wine and brandy. From molasses, rum is obtained. Now see, depending upon the raw material, the name of the alcoholic beverage is also differing. If it is rye, it is gin. If it is fermented rice, sake. Fermented potato, vodka. Fruit juice, then wine or brandy. Molasses, then rum. If it is barley, then beer. So depending upon the type of raw material, the alcoholic beverage will change. And again, depending upon the process of fermentation, again we can divide the alcoholic beverages into two types. We will see that also. Depending on the processing, we divide alcoholic beverages into two types. One is called as non-distilled beverage, and the second one is distilled beverages. Now, who are used in making this alcohol? Yeast is used in making alcohol. Alcohol is hazardous. It is injurious to health. Now, the yeast is only producing alcohol, and yeast also will die when the fermentation content, when the alcohol content in the fermenter reaches about 13%. Now, that means the natural alcohol what yeast has produced will be 13% only. Now, that 13% alcohol if you are releasing, then that is called non-distilled beverage. Example for non-distilled beverages are, we can take beer and wine as example. Beer and wine are examples of non-distilled beverages. 3 to 6% alcohol will be there, and 10 to 13% alcohol will be there in beer and wine. So alcohol content is less because distillation is not done. But when you come to distilled beverages, here they'll try to increase the alcohol content by doing distillation, by evaporation. So they increase the alcohol content. All the remaining like vodka, sake, gin, rum, all these things are examples of distilled beverages. If you see brandy and whiskey, you know, brandy and whiskey, they contain alcohol content of 40 to 60%. then rum and gin, the alcohol content here will be 40%. If we take vodka, vodka contains 60 to 80% of alcohol. So this much injurious it is. As the alcohol content increases, that much injurious it is. Okay. So this is about beverage industry. It is a fermentation process. The microorganism which is used is Saccharomyces cerevisiae only, and that is why it is called as brewer's yeast also. It will convert the sugars into alcohol and carbon dioxide, and depending upon the raw material, so these are the products we get. Depending upon the process, whether it is distilled beverage or non-distilled beverage, these are the examples. Take a screenshot, then we will talk about antibiotic production. Right. Now, the second industrial product is, we finished beverage, now we have to talk about antibiotic production. You know what is the meaning of antibiotic and who is the scientist who coined the term antibiotic? Waxman is a person who coined the term antibiotic. What's the person name who coined the term antibiotic? Waxman coined the term antibiotic. You know what is the meaning of antibiotic in Greek? The meaning of anti means against, right? And bio means life. Now, antibiotics, can you manufacture them in the lab? No. Antibiotics are chemical substances which are extracted from bacteria, which are extracted from prokaryotic organisms only. The only prokaryotes are bacteria, monomerans, right? So antibiotics, what are they? They are chemical substances which are extracted from prokaryotic organisms which are against the life of other organisms. Which are against the life of other organisms. They can kill disease causing organisms. From where are you extracting it, isolating it? You are isolating it only from prokaryotic organisms, monomerans. So for a pathogen, antibiotic is against the life, but for us it is saving our life. For us it is pro-life. For human it is pro-life. It is not killing us, no. So it is not anti-life for us. For us it is pro-life. And who discovered the first antibiotic? Alexander Fleming discovered the first antibiotic. What is the name he has given? He extracted that antibiotic from a fungus, from a mold. The name of the fungus is Penicillium notatum. That is why after the mold he named it as penicillin, right? Actually it is an accidental discovery. He was trying to grow bacterial colonies, Staphylococcus colonies he is trying to grow, but those petri dishes were infected, were contaminated with fungus Penicillium notatum, and this Penicillium notatum is releasing penicillin. That penicillin antibiotic is killing the bacteria. That's why bacteria are not able to grow. So like this he discovered it. So we can write on in 1928. In 1928, Sir Alexander Fleming. In 1928, Sir Alexander Fleming, he was checking Staphylococcus colonies. He is checking bacteria. The name of the bacteria is Staphylococcus. Staphylococcus means irregular bunch, like a grey bunch of colonies they are. So he is growing Staphylococcus aureus colonies in a petri dish, and this bacteria is not able to grow. Why the bacteria are not able to grow means the petri dish is contaminated with the fungi. What is the name of the fungi? The name of the fungi is Penicillium notatum. The name of the fungus is Penicillium notatum. What is it? It is fungi. It is a mold. Now this Penicillium notatum is producing an antibiotic called penicillin. This penicillin antibiotic is killing the bacteria. So that's why the bacterial colonies are not growing. So this was an accidental discovery discovered by Alexander Fleming in the year 1922. But its full potency, its chemical structure, that penicillin contains a beta-lactam ring. It is a broad spectral antibiotic. That complete structure was given by Ernst Chain and Howard Florey. Okay, but the complete structure was given by Ernst Chain and Howard Florey, right? So that's why these three people, Alexander Fleming, Florey and Chain, all the three people, they shared Nobel Prize in the year 1945 for discovering penicillin, for giving the complete structure and details about penicillin. These three people were awarded Nobel Prize in which year? In the year 1945. And this wonder drug, broad spectral antibiotic penicillin, is used to treat the soldiers during the Second World War, where many people got injured. This medicine helped them to recover nicely. Understood, children, about antibiotics? Now this was the discovery, right? Now how many antibiotics are there now in the market? You know, near about 7,000 antibiotics are there in the market now. 7,000 antibiotics are available in the market now. Now we cannot assume our life without antibiotics, so we are so much dependent upon antibiotics. And dangerous diseases like leprosy, diphtheria, plague and all, whooping cough and all, we are able to eliminate them by using microorganisms only. We are able to control them by using microorganisms only, right? Now in NCERT they asked us, can you list down some more antibiotics and from where are they extracted and what is the mode of action of that antibiotic, they asked us. So if you have done with this antibiotic, who coined the term antibiotic? Waxman coined the term antibiotic. What is the meaning of antibiotic? This word has been derived from a Greek word. Anti means against, bio means life, means bacteria will produce this antibiotic which is against the life of pathogens, which will kill the pathogens, but for us it is pro-life. Who discovered antibiotic? The first antibiotic was discovered by Alexander Fleming in the year 1928. He was growing a bacterial colonies, Staphylococcus colonies, which were not able to grow because the petri dish was contaminated with a fungus. The name of the fungus is Penicillium notatum, which is producing an antibiotic called penicillin, which is killing this bacteria. Now afterwards the complete structure and the details about the antibiotic were given by Chain and Florey. So Fleming, Florey and Chain shared Nobel Prize for their discovery in the year 1945. And now how many antibiotics are there available in the market? 7,000 antibiotics are there in the market. With these antibiotics doctors are able to treat so many disorders. Take a screenshot. Right, now let us list down some more antibiotics and from which organisms they are extracted and what is the mode of action of those antibiotics also. Since NCERT they asked us, so we have to make a list. So the name of the antibiotic we'll write here and from which microorganism it's extracted we'll write here and what is the mode of action we'll write here. Now we'll start with the fungal antibiotics first. Now penicillin is a fungal antibiotic only and this penicillin is extracted from a fungus called Penicillium notatum. Penicillin is extracted from a fungus called Penicillium notatum by Alexander Fleming. What is the mode of action of penicillin? How is it killing bacteria? Penicillin, what it will do? It will inhibit the cell wall synthesis. What the penicillin will do? When the bacteria is multiplying, new daughter bacteria has to make its cell wall. If new cell wall is not made, so the bacteria is not growing, then bacteria cannot survive. The mode of action of penicillin is it stops the cell wall synthesis of the bacteria. It inhibits cell wall synthesis. In this manner, it controls the pathogen. Next antibiotic we can take cephalosporin, extracted from a fungus called Cephalosporium acrimonium, which also has the same mode of action, inhibiting the cell wall synthesis. What is the second antibiotic? Cephalosporin, extracted from a fungus called Cephalosporium acrimonium. Now the mode of action, inhibiting the cell wall synthesis only. The third antibiotic is griseofulvin, extracted from Penicillium griseofulvum. So what it will do? It will inhibit cell membrane. Penicillium griseofulvum, it is a fungicide, means it can kill other fungi. So let us write down the third antibiotic. Griseofulvin, extracted from Penicillium Griseofulvum. And what the mode of action? It is a fungicide. It kills fungi. Right? It is a fungicide. So these three are examples for bacterial antibiotics. Now let us see the fungal antibiotics. Now let us see the examples of bacterial antibiotics. The first bacterial antibiotic is streptomycin. Streptomycin is the first antibiotic extracted from a bacteria called Streptomyces griseus, and this streptomycin was isolated by a scientist called Waxman, right? Let us see what's the name of the antibiotic. I told it is streptomycin. When we go and complain to the doctor about throat pain, they'll give us this antibiotic only, streptomycin. Streptomycin is extracted from a bacteria, actinomycetes member called Streptomyces griseus. Streptomyces griseus. And what is the mode of action? It stops protein synthesis. Where will protein synthesis happens? On the ribosome. Bacterial ribosome has a bigger subunit and a smaller subunit. Bigger subunit is 50S and the smaller ribosomal subunit is 30S. Now, what is the streptomycin action? It blocks, it inhibits the 50S ribosome formation, 30S. So, it inhibits 30S ribosome formation. So since it is not allowing the 30th subunit to be made, protein synthesis stops. So it blocks protein synthesis then. It will block protein synthesis then. So this is the action of one bacterial antibiotic. What is it called? It is called streptomycin. Another antibiotic is there. It is called oxytetracycline, which is extracted from a bacteria called Streptomyces rimosus. What is the name of the antibiotic? It is oxytetracycline, extracted from a bacteria called Streptomyces rimosus. Also has the same action, inhibits the 30th ribosome formation and thus blocks the protein synthesis. So it also has the same action, blocks the 30th ribosomal formation. Now next antibiotics, if you see bacitracin, extracted from Bacillus subtilis, what it will do? Another antibiotic, bacitracin, extracted from Bacillus subtilis. Now, like penicillin, what this antibiotic will do? So it will disturb the cell wall formation. So means it is inhibiting the cell wall formation. The same thing, it disrupts cell wall formation, similar to penicillin action. What is the name of this antibiotic? Bacitracin. Another antibiotic is there called polymyxine, extracted from Bacillus polymyxa. Now what this will do? If bacitracin is inhibiting cell wall formation, polymyxine will inhibit cell membrane formation. It disturbs cell membrane formation. It disturbs cell membrane formation. Next antibiotic is erythromycin, extracted from Streptomyces erythraeus. So what it will do? Next antibiotic is erythromycin, extracted from an extract from bacteria called Streptomyces erythraeus, T-R-E-U-S, Streptomyces erythraeus. It will stop the 50S formation of the ribosome. It inhibits 50S ribosome formation. That means it is also blocking protein synthesis. This is also blocking protein synthesis. Enough children, these many antibiotics. So in NCERT they asked us to collect some more antibiotics. Yeah, we collected fungal antibiotics, we collected bacterial antibiotics, and from where are they extracted, from which microorganisms they are extracted, and what is the mode of action. Take a screenshot, then we will talk about the production of certain organic acids, enzymes and bioactive amines, which also comes under the same side heading 10.2. Take a screenshot fast. Yes. Now, under this, we are going to talk about organic acid production, enzymes production, and then bioactive amine production also. Now, microorganisms. In our chemistry labs, we will be using acids. Those acids are extracted from microorganisms only. What are the organic acids which are extracted from microorganisms? I will tell you easy method. So write an acid with A, write an acid with B, write an acid with C. Acid with A is acetic acid, acid with B is butyric acid, acid with C is citric acid. So acetic acid, butyric acid, ABC, citric acid. Now, acetic acid is obtained from Aspergillus niger. Acetic acid is obtained from Aspergillus niger, which is a fungus. Butyric acid is obtained from Clostridium botulinum, which is a bacteria. Right? And citric acid is obtained from Aspergillus. Citric acid is obtained from Aspergillus niger. I'm sorry. Acetic acid is obtained from Acetobacter acetium. Acetic acid is obtained from Acetobacter aceti. Citric acid is obtained from fungus which is called Aspergillus niger. So we have seen, and the fourth acid what we already know is lactic acid during curd formation used by a bacteria called Lactobacilli. So these are the acids, and where do we use it? We use them in chemical labs as solvents. So we use them in reagent preparations, and we use them as preservatives also. In packing the foods and all, we use them as preservatives. So these are acids A, B, C: acetic acid, butyric acid, citric acid, and lactic acid. Acetic acid, Acetobacter aceti. Butyric acid, Clostridium botulinum. Citric acid from Aspergillus niger. Lactic acid from Lactobacilli. Now let us talk about enzymes then. Now there are so many enzymes. We will see two main enzymes. The first enzyme is lipases. Now from bacteria we extract lipase enzyme. Where do we use this lipase enzyme? In laundry industry, in dry cleaning industry, to remove the oil stains. So in detergent formulations we use this lipase. We extract lipases from microbes, from microorganisms like bacteria. We extract lipases, and where do we use this lipase? In detergent formulations. We use it in dry cleaning industry. Why do we use it in dry cleaning industry? To remove the oil stains. To remove oil stains. So this is one enzyme. And coming to the second enzyme, they are called proteases and pectinases. From bacteria we extract proteases and pectinases. Now if we prepare a fruit juice at our home, it will be brisky, it will be like pulpy, right? It will not be appealing to drink. But when we buy a same bottled beverage, bottled drink, when we buy bottled fruit juice, we'll buy, it will be clear. That means they'll add these proteases and pectinases to clarify the bottled fruit juices, so they'll be appealing to drink. So proteases and pectinases, where do we use? We use them for preparing bottled juices, bottled fruit juices, to clarify the fruit juice. To clarify the fruit juice, they add proteases and pectinases. So we finished enzymes, we finished organic acids. Now the third enzyme is there, which is called streptokinase. What is the third enzyme? Streptokinase is an enzyme. Streptokinase is extracted from a bacteria called Streptococcus. It is extracted from bacteria called Streptococcus. Now why are we talking about this enzyme streptokinase? You know, it acts as a clot buster. It acts as a clot buster. Now when in the arteries and veins, if cholesterol goes and deposits and it makes clots, it leads to myocardial infarctions, it leads to heart attacks, right? So now then what will the cardiologist do? They'll do angioplasty and then they blast those cholesterol esters and all. Now instead of doing that painful process from the nostril and all, they will send the tube into the heart, they will locate it and they'll blast it. Instead we can just take an enzyme called streptokinase which acts as a clot buster. So what it will do? It will dissolve clots present in the arteries and the veins. It will dissolve blood clots present in the arteries and veins, and it prevents myocardial infarction. It prevents myocardial infarction. Thus it prevents heart attacks, right? Thus it prevents heart attacks. See how important, how wonderful enzyme it is, streptokinase. Now take a screenshot. There are 2 bioactive amines that also we will talk about. Right? So now coming to bioactive amines, there are 2 bioactive amines. One is called cyclosporin A, and we'll write down bioactive amines. Under bioactive amines, the first one is called as cyclosporin A. What is the action of cyclosporin A? It acts as an immunosuppressive agent. It will suppress the immune system. It acts as an immunosuppressive agent and it is extracted from a fungi. The name of the fungi is Trichoderma polysporum. It is extracted from a fungi called Trichoderma polysporum. So when do we use immunosuppressive agents? Means when a person has undergone transplantation, from the donor we are bringing the organ we are transplanting here. Now this person's immune system should not identify that organ as a foreign organ. It should not damage it. So that is why for the person who has undergone transplantation, the doctor will give immunosuppressive agents so that the organ is safe in his body. Now example for the immunosuppressive agent is cyclosporin A, extracted from a fungus called Trichoderma polysporum. So for whom do we give it? For a person who has undergone transplantation of organs. So this is one bioactive amine, and the last bioactive amine is statin. It is statin. Statin is extracted from an yeast. It is also fungi only. Monoecious purpureus. It is extracted from an yeast called Monoecious purpureus. What is this statin called as? It acts as a blood cholesterol lowering agent. Statin acts as a blood cholesterol lowering agent. You know, our blood should contain 150 to 250 mg cholesterol. Our blood should contain 150 to 250 mg cholesterol per deciliter of blood, per millimeter of blood. Now, if the cholesterol levels are more than that, then it is wrong. It's dangerous. So then, if we take a pill called statin, what it will do? It will decrease the blood cholesterol levels. It will decrease the blood cholesterol levels. It will stop the further cholesterol synthesis. This statin acts as a competitive inhibitor. Okay, the statin acts as a competitive inhibitor. It stops the cholesterol synthesis. It acts as a competitive inhibitor for an enzyme called HMG-CoA synthase. Now this statin comes and binds to HMG-CoA synthase. It will not allow the HMG-CoA synthase to do the cholesterol synthesis. Done. That's it. So then the cholesterol will not be made in our body. Then a person is safe. The person will not get any heart attacks. Right, children? So what did we discuss under 10.2 microbes in industrial products? We discussed about beverage industry, brandy, whiskey, rum, all those things we discussed. Then we came to antibiotics, penicillin, erythromycin, streptomycin, oxytetracycline, all that we discussed. Then we came to enzyme production, organic acid production, and bioactive amines. Streptokinase is one bioactive amine, cyclosporin is another bioactive amine, statin is another bioactive amine. With this, we completed 10.2. Take a screenshot. We'll talk about 10.3, microorganisms in sewage treatment. Now we'll talk about the next topic, that is microorganisms in sewage treatment. Now we need to understand here what is sewage. Municipal wastewater is sewage. Now what is sewage? Sewage means it is municipal wastewater. Now what does the municipal wastewater contain? It contains 95.5% of water only, in which the organic and inorganic matter will be there with the pathogenic bacteria and fungi also. So sewage means municipal wastewater. What does the municipal wastewater contain? The municipal wastewater contains 95.5% of water with organic and inorganic matter. It contains organic matter and inorganic matter with pathogenic bacteria and fungi are also there in this sewage, with pathogenic bacteria and pathogenic fungi. Since disease causing bacteria and fungi are there, so we should treat the sewage before releasing it into an open land or into a water body. And you know, the main component of the sewage is human excretory material. So what is the main component of sewage? You know, the main component of sewage is human excreta. Now in our elementary canal only all the pathogenic organisms will be there. That pathogenic organisms will be released into the human excretory material. Now that pathogenic organisms, if we don't treat it, then what happens? Tell me. If we don't treat it, then it causes outbreak of waterborne diseases, right? If the sewage, if it is untreated, it causes outbreak of waterborne diseases. It causes outbreak of waterborne diseases. Can you name few waterborne diseases? Typhoid is a waterborne disease. Jaundice, typhoid we can tell, jaundice we can tell, cholera we can tell, dysentery, all these are waterborne diseases. Waterborne diseases examples I am writing out here: typhoid, cholera, jaundice, dysentery, all these are waterborne diseases. Outbreak of waterborne diseases will happen, so if we don't treat the sewage. Next thing, the water bodies are also getting polluted. That's the second drawback. Even the water bodies are polluted. Without treating, if you dump that sewage in the water bodies will get polluted. That's the second drawback. Now this polluted water bodies start giving foul smell and all, they damage the environment. So these water bodies, they give foul smell and they kill the fish and all, and it chokes the water bodies to death. So this pollution, this water pollution will finally choke the water body to death. The water body will die, which is not at all good. So these many disadvantages will happen if we don't treat the sewage. So that means the sewage should be treated. Where will we treat the sewage? In a particular plant which is called as STP, sewage treatment plants. So the sewage is treated. Where do we treat it? In STPs. What do you mean by STPs? Sewage treatment plants. So the sewage is treated in sewage treatment plants. We treat the sewage in sewage treatment plants. Understood till here? Now, this is introduction to sewage. Take a screenshot, then we will talk about the steps of sewage treatment. Right, now how many steps are there in sewage treatment and who are involved in decomposing the sewage? You know, so man has designed robots, man has designed rockets, man has went to other planets also, but for decomposition of sewage from long back onwards we are using microbial technology only. Means so far we were not able to prepare any efficient technology which is better than microbial technology. Means microorganisms are only used to treat the sewage. Okay, so microbial treatment we do to decompose the sewage. To decompose the sewage, we go with microbial treatment only. Now, so far, there's no man-made technology which could beat the microbial technology. How many steps are there in sewage treatment? There are 2 steps, primary treatment and secondary treatment. So, we have 2 steps of sewage treatment. The first one is called as primary treatment. The next one is called as secondary treatment. Primary treatment involves physical removal of the floating particles and the sedimenting particles. So that is why it is also called physical process. Since microorganisms are not involved here, so we also call it as an abiological process. So what are the other names we can give for primary treatment? Means since it involves a physical removal of the floating and sedimenting particles, we call it as a physical process also. It is also called physical process. Since microorganisms are not involved, it is also called as a biological process. Whereas in secondary treatment, microorganisms act to decompose the sewage. They release enzymes and they start decomposing the sewage by aerobic treatment and anaerobic treatment. So secondary treatment is also called as biological process. Secondary treatment is called biological process. It is called biological treatment. Understood till here? Now, what do we do in primary treatment? We remove the suspended particles which are suspending, which are floating, and we remove the sedimenting particles in primary treatment. The suspended and sedimentary means bottom settled particles. Sedimentary particles are removed here. Whereas in secondary treatment we are using microorganisms to decompose the organic matter. So here we do sedimentation and suspension particles are removed. Here what do we do? The organic matter which is present in the sewage will be degraded by microorganisms. In biological treatment, the organic matter is decomposed by microbes. Right, now again if we see under primary treatment how many steps are there and under secondary treatment how many steps are there. So primary treatment proceeds with 2 steps. Now what are the 2 steps of primary treatment? I told if any floating particles are there, so the floating suspended particles are removed by filtration. Now under primary treatment the first step goes is filtration. Now what do we do in filtration? We pass sequential filters, 7 layers of sequential filters through which the sewage as it comes down, different floating particles will be removed off, collected off. In filtration, 7 layers of sequential filtration is done. 7 layers of sequential filtration is done to remove all the floating debris. Which type of floating debris we expect in the sewage? Water bottles, waste bottles, paper, all that waste, floating debris will be removed by filtration step. Now the second step is called as sedimentation. Now what do we do in sedimentation? In sedimentation, grit is removed. Grit means it includes the small stones, sand, right, pebbles. All these things, due to their gravity, they settle to the bottom. So those things are removed in sedimentation. Now if you see this sedimentation part, all those sand, the stones and all, pebbles and all will get collected at the bottom here. And then whatever the liquid which goes on the top, what is this liquid called? This liquid is called as primary effluent, whereas this bottom settled sedimented mass is called as primary sludge. What is this called? This is called primary sludge, which includes stones, pebbles, sand and all the sedimented particles. Now this primary sludge, what do? We take this primary sludge, and this primary sludge can be dried and it can be used for landfilling. It can be dried and it can be, because it is so stone and sand only, it can be used for landfilling, or it can be used for compost. It can be used for composting also. Now we are going to take this liquid part which is called as primary effluent. This primary effluent is sent into the secondary treatment. This primary effluent is sent into the secondary treatment. Now take this primary effluent part and come on here. Take this liquid part and come on here. Now in secondary treatment, so we have 3 stages again. What are the 3 stages of secondary treatment? If we have to see the 3 steps of secondary treatment, the first one is called as, what is the first one called? Aeration tank. So if I write 1 and 2, 3, 4, 5, I will write here. So the third one is called as aeration tank. Fourth one will be called as settling tank. And the last one, fifth one, will be called as anaerobic sludge digester. It is called as anaerobic sludge digester. Now they will give all these 5 and they'll ask you to arrange them in order, so that's why I try to keep them in line. First filtration, followed by sedimentation, then in aeration tank, then settling tank, then anaerobic sludge digester. We took all this liquid here which is called as primary effluent, and we brought the primary effluent to a big tank which is called as aeration tank. Now what do we do in this aeration tank? This is the aeration tank. In this aeration tank, take all the liquid. All this liquid is there. Now this liquid should be mixed nicely. So means keep a rod, means agitation system. This is agitating it. And what does this liquid contains? This liquid contains organic matter. This liquid contains organic matter. It contains bacteria and it contains fungi. Food is there, heterotrophic bacteria and fungi are also there in this primary effluent. And aeration you are doing, agitation you are doing. At this point, if you check the BOD, you should understand now what is BOD here, biological oxygen demand. If you check the BOD here, the BOD of this untreated sewage will be very high. The BOD now, it will be very high. It will be something around more than 1600 mg oxygen per liter. This much amount of BOD and high indicates highly polluted body, right? Since we didn't start the process, the BOD will be very high, biological oxygen demand. Now what happens here means the bacteria and fungi, they start decomposing the organic matter in the presence of oxygen. So oxygen is there, so that's why it is aeration tank. They start decomposing the organic matter, right? So when they start decomposing the organic matter, then they get the food, then they grow nicely and they make flocks. What do you mean by flocks? Means the fungal filaments will be like this, and in between these fungal filaments the bacterial cells will get trapped. In between the fungal filaments, these are the bacterial cells, they get trapped. Now it makes a mesh-like structure. Now this mesh-like structure is called as floc. So microorganisms, heterotrophic microorganisms like bacteria and fungi, they will make flocs here. Now these flocs, they secrete enzymes and they start decomposing the organic matter. Now when they are growing, they require more oxygen, so they consume more oxygen. So when they consume more oxygen, when they demand more oxygen, when the organic matter is there, so that means more the pollution, more the organic matter, more the oxygen consumption. So that is why we'll tell the oxygen demand of the polluted body will be more. As they are eating off, what happens? The BOD starts decreasing and the BOD gets reduced to 10 to 15% of the original sample. When the BOD drastically reduced, means oxygen is not demanded by the bacteria. Why is oxygen not demanded by the bacteria? Means there is no organic matter there. That is why they are not growing. If they are not growing, they will not consume oxygen. So if they are not growing, means organic matter is not there, means the sewage has undergone decomposition. It got clear now. So now at that stage we will take all this and we will go to the next tank, which is called a settling tank. I think you understood now. Under secondary treatment, the first step, which is called as aeration tank, we aerate it, we agitate it, and the heterotrophic bacteria and fungi grow nicely and they make the flocs. These flocs, they consume the organic matter and they decrease the BOD. Okay, if you want the definition of BOD, it refers to the amount of oxygen consumed. It refers to the amount of oxygen consumed by the bacteria in 1 liter. In 1 liter of sewage, right? In 1 liter of sewage, how much oxygen is consumed by the bacteria to decompose the organic matter? That is a definition of BOD. And above this, it is highly polluted BOD. If it is 1400, 1500, it is moderate. If it is less than 1400, it is a weak sewage sample. Okay. Now when the BOD drops down, we will take it to settling tank. Now we will take all this to settling tank. Now in the settling tank, if we go, now here they have rigorously grown, they have rigorously acted. Now take all this matter here. Now here it is, we are asking them to settle now. We are asking them to take rest now. They will settle. No aeration, no agitation, they are settling now. Now this settling liquid, what is it called? It is called activated sludge. In settling tank, what do we have this? This is called activated sludge. This liquid which is settling now, which is rich of bacterial flocs, is called as activated sludge. It will settle nicely. So when the liquid comes here, that means this becomes vacant. This becomes vacant. When the container becomes vacant, the person will drop new sewage here. The person will drop new sewage here. When the new sewage is coming into this container, now again bacteria should start the process. Now if the bacteria has to grow inside this, it will take time. The bacteria are here, right? The bacteria are here. Why can't we pump little bit of the activated sludge from here to here, so that which acts as an inoculum and start the next round of process faster? They do that. When we take all this green colored content into the settling tank, little bit of the activated sludge is pumped back, which acts as an inoculum to treat the next slot. And the remaining activated sludge, now it is taken to the next step, which is called as anaerobic sludge digester. The remaining activated sludge is now taken into a next one, which is called as anaerobic sludge digester. If we have to see the picture of anaerobic sludge digester, it is also a big container. See, since it is aeration tank, we kept it open. Here also it container is open, container is open, container is open. But here it is anaerobic, means oxygen should not be there, so seal it, no. Then it becomes anaerobic. Now this is called anaerobic sludge digester. What do we do here in anaerobic sludge digester? In anaerobic sludge digester, now take all this activated sludge. Take all the activated sludge into this anaerobic sludge digester. Now in this anaerobic sludge digester, under anaerobic conditions, another set of bacteria will act. Now under aerobic conditions, aerobes acted, no, heterotrophic bacteria and fungi. Now when you maintain anaerobic conditions here, anaerobic bacteria will act, methanogens will act. So are you adding any methanogens here, or they are also present in the cow dung only? They are also present in the sewage only. They are present in the sewage only. But when the conditions becomes favorable, then they start working. Now under this tank, what happens? Anaerobic methanogenic bacteria starts working here. What do? Anaerobic digestion they do. They convert the leftover little bit of remaining materials into, what do they convert? They convert them into methane, hydrogen sulfide and carbon dioxide. They do fermentation, they do methanogenesis, and they make methane, which is a major component of biogas. It is a major component of biogas. After anaerobic sludge digestion, what do we get? We get biogas and we get the remaining material. In this, the liquid portion is called as secondary effluent. This liquid portion, what is it called? It is called secondary effluent. The secondary effluent, you can freely release it into the water bodies. The secondary effluent, freely you can release it into the water bodies. It doesn't have any pathogenic organism. So after the secondary effluent is released into the water bodies, the leftover solid gas is collected, liquid is thrown into the water bodies. Now because it is treated, not an issue. The solid which remains here acts as a compost. The solid which remains here acts as a compost. See, by doing sewage treatment, how many products are we getting? Here we are getting compost, which is used for landfilling, and next here you are getting biogas. Then this water is released into the water bodies. You can use it for irrigation purposes, you can use it for giving water for the animals and all. You can use this water, and the left out solid, secondary sludge. Secondary effluent is a water. Secondary sludge can be used as compost. This is a treatment of sewage, which is done in sewage treatment plants by the municipal organizations. Now by the Ministry of Environment and Forest, the Indian government has initiated to purify our sacred rivers. Like Ganga Action Plan they have initiated, Yamuna Action Plan they have initiated, because as the urbanization is increasing, now the municipalities are not sufficient to treat the sewage plants and all. In villages and all, without proper treatment, all the sewage has been dumped into the water bodies, and all the water bodies will mix into the rivers and lakes. So then our sacred rivers are getting polluted. So the Ministry of Environment and Forest, they have initiated a huge project. They have spent lakhs and lakhs of money there to purify our natural rivers. Like Ganga Action Plan and Yamuna Action Plan, it's not a small step process. If it is a small water tank in your house, you can clean it, but it is a river which is flowing through many states and it is coming. How do you purify it? Now at every 1 kilometer, so you have to take all the water out, you have to drain the water from there and do primary treatment, secondary treatment, and after treating, collect the secondary effluent and release it back. Now it mixes with the already floating water here. Now from here it goes 1 more kilometer. Again there you collect it, and from there again you do primary treatment, secondary treatment. After treating it, collect that secondary effluent and mix it with the water. So like this they have done at a stretch of 1 kilometer, and for 1 year or 2 years they have done it, and they have spent so much money to treat and to bring back our natural rivers. With this we finish sewage treatment. 10.4 Microorganisms in biogas production we will discuss next. Take a screenshot. The next one is microorganisms in biogas production. It is 10.4 microorganisms in biogas production. So to save the time, I already took everything on the board so we can see here. Now this is a typical biogas plant. This is a typical biogas plant, and who developed this biogas production means there are two organizations. One is IARI and the other one is KVIC. They developed this biogas technology and they introduced it in villages where cow dung cattle is there. Now what are the full forms of IARI and KVIC? IARI stands for Indian Agricultural Research Institute. Indian Agricultural Research Institute and Khadi Village Industries Commission. Khadi Village Industries Commission has developed the biogas technology and they introduced it in the villages. Earlier, people in villages used to use cow dung cakes and all, wooden logs and all to make to generate that flame to cook those things. Now with this biogas, you know, their health is improved, the environment is saved. So how to produce biogas production? Let us see. This I told you is a typical biogas plant, in which if you see, this is the main digester. This is a main digester into which the sample should be sent. Now here I wrote the numbers 1, 2, 3, 4 and this is 5 and here is 6. Now the first one is called as mixing tank. Cow dung or whatever is the waste raw material, wooden straw you can take, or the cattle dung we can take, cellulosic material we can take, potato straw, all that we can take. We can take the raw material, mix it with the water nicely and prepare a thick slurry, uniform slurry. Since we are mixing the contents here, the container is called as mixing tank. In NCERT they have given this. Now here you have a thing here, so when we close it, then only you can mix the compartments here. When the slurry is ready, then we'll open this valve and the slurry is sent through this one which is called as inlet chamber. The slurry comes through the inlet chamber into the third one which is the main digester tank. So the slurry now comes here. Don't overdump the digester, only 2/3 of the digester, only to the 2/3 of the digester level we will take the slurry. Why are we taking only to the 2/3 level? Means since it generates gas and for the gas we need space, so that is why to 2/3 level only we will take the slurry, and it is fermentative process, anaerobic process, so we will ensure anaerobic conditions. It is an airtight chamber. The typical dimension of a digester is, it is 10 to 15 feet concrete tank. In NCERT they have given the typical biogas plant consists of a digester. This is a digester which is 10 to 15 feet deep. It is concrete cylindrical tank. You can see here it is cylindrical, it is concrete, and it's an airtight chamber in which the cow dung and water is there. Now when we talk about the cow, goat and all the cattle, in their alimentary canal they have an additional part called rumen. In the stomach they have an additional part called rumen. Now this rumen of the cattle, they host symbiotic bacteria. Which type of symbiotic bacteria present in the rumen of the ruminating animals like cattle and all? They are called methanogens, methanogenic bacteria, methanococcus, methanobacter, right? All these bacteria will be there. Now those bacteria which are already present in the cow dung, under favorable conditions they start fermentation. They do methanogenesis, they generate biogas. As the biogas gets generated, now there is a cylinder here which is called as gas holder. The gas gets collected here, and when the gas gets collected, the floating cover, you know, it starts hitting, it starts making sound because the gas is coming and collecting here. It gets weighted and then it starts hitting up, so that is why it is called gas holder, it's called floating cover. When the gas gets collected here you have an outlet. Through this we can take out the gas, we can use it for various purposes, inflammation purposes, or for electrical in agriculture for pump set generation and all, lighting purposes. We can use this gas which is called as biogas. Now what happens to this leftover waste material? Now there is one valve here at the inlet chamber which is called inlet valve, and there is outlet valve. I told you, you need to maintain it as an anaerobic chamber. You have to ensure anaerobic conditions, so that's why we keep valves here and to try to maintain it as an anaerobic condition. Now when we open the outlet valve, then the waste material goes through the outlet flow. This is inlet chamber and this is the outlet chamber. It gets collected here. All the waste gets collected here. Now what is this is called? This is called overflow tank. Here it gets collected. Now in the overflow tank we dry that material. After drying it up, so we can use it in our agricultural fields. It acts as a good manure. This one which you are drying up acts as a good manure. See, you started with a waste material only, cow dung, which can also be used as a material. From that that waste we generated biogas, and after generating biogas also still you are getting that waste which you are drying out and you are using as a manure in agricultural field. Means zero waste, right? Everything has been used here. So it is such a good technology which was developed by Indian Agricultural Research Institute and Khadi Village Industry Commission. So I think we understood the process of biogas. Now let us see what is the information and how many steps are there in biogas production. Now biogas is a combustible gas. Yeah, it is a combustible gas only which is produced by anaerobic process only. This is an airtight digester, so it is produced by anaerobic breakdown of the organic matter which is present in the cow dung. So this point we have already seen the typical biogas plant. What are the dimensions? It is 10 to 15 feet, cylindrical, it is a concrete tank, right? Now biogas production occurs in 3 stages. The first stage is called as solubilization, the second stage is called as acidogenesis, and the third stage is the exact process where methanogenic bacteria acts and biogas is produced is called as methanogenesis. The 3 steps of biogas production: first one is called as solubilization or decomposition, second one is called as acidogenesis, and the third one is called as methanogenesis. Now we'll start the first step which is called decomposition or solubilization. Now in decomposition or solubilization, facultative anaerobic microorganisms, if we have to take the examples for facultative anaerobic microorganisms, they are Pseudomonas and Clostridium. They act on the raw material, cellulose, they act on the cow dung, and they secrete enzymes, to decompose the raw material. What are the enzymes they secrete? They secrete cellulases, lipases, proteases, pectinases. They are trying to convert the complex organic materials into simpler forms, into monomeric units. Who will do? Decomposing bacteria, facultative anaerobic bacteria like Pseudomonas and Clostridium will do, they'll convert them into simpler monomers. This is the first step. Then comes the second step which is called as acidogenesis. In acidogenesis, another set of bacteria like Propionibacterium aceti, what do? They convert the monomeric forms, they convert the simpler forms into their respective acids, into organic acids. Since from the monomers, from the simple organic compounds, acids are getting generated, so the process is called as acidogenesis. So fermentative microbes like Propionibacterium aceti and all, they convert these simpler monomeric compounds into their respective acids. Formic acid, like that they generate acids. Now that's why the process is called acidogenesis. The third step, the actual step, which is called as methanogenesis. Here the methanogenic bacteria, they are archaebacteria only. Archaebacterium, methanobacterium like Methanococcus, methanobacteria and all, they act on the organic acid and they generate methane, H2S, and they generate carbon dioxide, all those things, sulfur they generate. So methanogenic bacteria like archaebacteria and methanobacterium, they convert these organic acids into methane, which is the main thing. See here in the collecting tank we have seen methane is there, carbon dioxide is there, oxygen can never come, it is an anaerobic process. So carbon dioxide will come, H2S will come, hydrogen will come, all that is the composition of the biogas, but the major composition, the major component will be methane. That is why it is called as methane gas or biogas. It's called gobar gas because gobar, cow dung, is used as a raw material. It's called as a marsh gas because we are taking the marshy compounds, right? So now talking about the advantages of biogas. Now instead of cow dung cakes, they are using this biogas which is cleaner and no foul smell, no smoke and all is generated. That's the second advantage. And the biogas is completely combustible gas, whereas the trees and all, the cow dung and all will not completely burn, means there's no wastage. And where can we use this biogas? We can use it for cooking purposes, for lighting the lamps and all, in pump sets, in agricultural areas we can use it. And then if any pathogenic organisms are present in the cow dung, so in this biogas even they will get killed off. All these are the advantages of biogas. What are the advantages? No smell, no smoke, complete combustion will happen, and from waste we are generating it, and the microorganisms present in the cow dung will also be killed off, and this biogas can be used for cooking, lighting and pump sets, all those things. Children, all this is about microorganisms involved in biogas, gobar gas production. Now we are left with the last two topics, microorganisms as biofertilizers and microorganisms as biocontrol agents. Take a screenshot, we will continue. So the next topic is microbes as biocontrol agents. So this is theoretical topic. I thought from these statements, statement based questions will come. From these sentences, statement based questions will come. So let us try to understand the NCERT sentences as it is, children. So that is why I took the NCERT sentences directly, and these are the examples at the end we'll see. So microbes as biocontrol agents. Now for example, earlier when there is no medicines and all, when this is not there, so we used to do home treatment, right? Taking ginger, taking garlic, or taking tulsi, all those things we used to do treatment by ourselves. But nowadays for anything we'll take an antibiotic, or we'll go visit the doctor and bring these many capsules and we'll try to heal our body. In the same manner, here also what happened, if you are having an agricultural farm, in an agricultural farm, if the farm is getting infected with a pest or with an insect, to control that pest and insect, nowadays the ready-made solution what you have is go to a fertilizer shop, chemical pesticide shop, buy a pesticide, insecticide, bring it, take it in a spray it and kill all of them. So it is a ready-made solution. But by doing this for over a period of time, what happened means the environment got damaged and the human health got deteriorated, because those chemicals and all are cancer causing. They will deteriorate the health, the first thing. Soil, water, air, everything got damaged, the second thing. Now there is a pressure on the farmers to switch to organic farming, to protect the environment, to protect the people's lives. So that's what they have given here. Now let us see what they have given here. Microorganisms as biocontrol agents. I told you, to control agricultural insects and pests, what is the ready-made solution we have is to go buy the chemical, like to kill the insect, insecticide, to kill the pest, pesticide. But by doing this, it is causing, these chemicals are extremely harmful to the human health. Not only to the human health, it is damaging our environment also. Like air is getting polluted, water is getting polluted, soil is getting polluted, right? So instead, biological control, instead of using insecticides and pesticides, if we use biological organisms to control insects and pests, that is called biological control, is found to be beneficial. Using, see, these are the examples. To control aphids, so you can use its predator called ladybird. Introduce ladybirds into your farm, they will find out the aphids and they'll eat off the aphids, right? And if you want to control mosquitoes in your agricultural farm, like rice plantation, wheat plantation, introduce dragonfly. This is the predator for mosquito. This is coming under biocontrol agents. Pests are controlled by introducing the predator organism. These are the predators which will control these pests. Ladybird is a predator for aphids, dragonfly is a predator for mosquitoes, right? Now this is the what organic farming means. This is what biocontrol means. An organic farmer believes that biodiversity will further the health. What do you mean by biodiversity? Having different organisms, right? If different organisms are there, then it is diversified. So if the ecosystem is diversified, then we are healthy, right? So more varieties on the landscape. Organic farmer believes that there are more varieties of organisms on the landscape, then that landscape will sustain for a longer period. That is what they believe, right? Therefore, an organic farmer works to create a system. He will work to create a system not to kill the insect, not to kill the pest, but he will try to keep them under control, not by spraying insecticide and pesticide, but by introducing their predators. So, organic farmer works to create a system where the insect or pests are not eradicated. You are not completely eradicating them. You are not killing them. Instead, they are kept at manageable levels of their systems. You will check it, you will balance their levels, and you will try to maintain a vibrant ecosystem. Means you are giving space for the organisms also to live. Even though they are disease causing, you are checking them so that they are not too much damaging your farmland. So in contrast, if you go with conventional farming, what we have done till now, using conventional farming, we use chemicals which will kill the insects. See, it will kill the beneficial insects also, it will kill the insect and pest also. So it doesn't have any differentiation that this is good, I should not kill it. This is a pest which is damaging the farm, I have to kill it. Invariably it will kill both the things. So in conventional farming, when we use chemicals, it will kill both the useful and the harmful lives indiscriminately. It will kill. This is a holistic approach that seeks to develop an understanding of the webs of interactions between the merits of the organisms that contribute to the field of flora and fauna. So you have to switch to organic farming. When you are switching to organic farming, you should have a complete understanding about the life cycle of the pest, about its habitat, and about which time it comes out, which type of fodder, which type of plants it eat, and what is its complete life cycle, and what are the interconnecting webs. You have to understand, then only you can go with organic farming, right? The organic farmer holds the view that eradication of pest is not completely possible. So you might bring an insecticide and kill all the insects in your area. Next day morning, from the surrounding city, they can flew and they can come and they can eat off your farm again. So if you are thinking that I can eradicate it completely, organic farmer's belief is correct only. He tells that you cannot completely eradicate all the insects and pests, but also undesirable. Eliminating them completely is undesirable. It is not good. For without them, the beneficial predators will not survive. If you are killing the insect is the food for a predator, and that predator is beneficial for us. You are killing the insect, then in turn the predator will not have the food, predator will die. So the predator will not survive, it cannot survive. So organic farmer, here they have told, like a biological control method is found to be beneficial, where if you want to control the pest, introduce the predator. These are the examples they have given in NCERT under biocontrol. Let us see the examples now. So children, these are statement based questions, so that is why I directly copied the sentences from NCERT. Hope you understood these sentences. Now let's go to the examples. Under examples, they have given the predator, like we know this ladybird which is having red colored patches, right, markings on its body. It's called ladybird. This is a predator which can eat off aphids. If you want to control aphids, so this is a serious pest for rice plantation, wheat plantation. If you want to control the aphids, then introduce ladybirds into the farm area. They will control this one. Then in the next manner, mosquitoes are also the pests. If we want to control the mosquitoes, then introduce dragonfly which will eat off this. And another one is praying mantis. This is not given in NCERT. Praying mantis. Praying mantis, it has got its name because it holds its legs like in a praying position. That is why it's called praying mantis. Praying mantis is a predator for wide variety of insects. This will eat only aphids, this will eat only mosquitoes. But praying mantis eats many varieties of insects. This also you can introduce it. Then coming to microbial control, so these are predator controls. Coming to microbial controls, so we are aware of a bacteria, I think it is called as BT, Bacillus thuringiensis. Bacillus thuringiensis produces a toxin which is called BT toxin. Now when the caterpillar comes to damage the crops, now when it feeds on the leaf sap, when it feeds on the sap, now what we need to do? Buy BT spores, they are available in the sachets in the market. Mix the BT spores in water and spray it on the agricultural land. Now BT spores are there, bacteria is there on the leaves. When the caterpillar comes and when it eats the leaf, when the caterpillar comes and eats the leaf, what happens? On this leaf, bacteria, Bacillus thuringiensis bacteria is there. This Bacillus thuringiensis bacteria will release BT toxin. The bacteria will produce a toxin called BT toxin. This BT toxin is taken up by this caterpillar into its alimentary canal. Now in the alimentary canal, in the midgut, foregut, midgut, hindgut, in the midgut of the caterpillar, this BT toxin becomes active. Why it becomes active? Till now it was inactive. That is why it's not damaging the crop. Once it goes to the caterpillar's alimentary canal, that too in the midgut, looking at the alkaline pH of the midgut, the inactive toxin becomes active toxin. Once when it becomes active, it goes and damages the intestine. It goes and binds to the epithelial cells of the intestine, causes pores on the intestine. If the caterpillar's intestine develops pores, if the caterpillar's intestine develops pores, can it survive? It cannot survive, so it will die. Now all the caterpillars are dying, or only those caterpillars which are damaging our crop are subject to death. Only those are subjected to that. This is what is biocontrol about. Means we will kill only those insects which are damaging the crop field, not all types of insects. This is better, right? So under microbial control, we talked about bacteria. It is called as Bacillus thuringiensis. It produces a toxin, Bt toxin. That Bt toxin, it's inactive normally. That's why it don't harm anyone. When the insect takes it in the midgut at the alkaline pH, it becomes active. It causes pores on the intestine and the caterpillar will die. Now let us talk about a fungus also, which acts as a biocontrol agent. The name of the fungus is Trichoderma. It is a free living soil fungi. It's a free living soil fungi. This fungi, it enters the roots of the plant. It enters the roots of the plant, and if the plant is with the Trichoderma fungi, it will not give place for any other root-borne pathogen to enter the plant. Good. If the plant is with the Trichoderma, then no other root-borne pathogen will dare to enter inside. So in that manner, it protects the plant. Now coming to virus, there is a virus which is called as baculovirus. This baculovirus, now this is even good. Baculovirus, under that there is a genus which is called NPV, nucleopolyhedrovirus. Now this nucleopolyhedrovirus is species specific. It's a very narrow spectral insecticide. Means only that particular type of insect only it will kill. It will not cause any harm to any other insects, not to the plant, not to the bird, not to the humans and animals. Means it's that much species specific, and it's very much narrow spectral antibiotic, and narrow spectral one baculovirus. It attacks insects and arthropods. Now where do we use this baculovirus? Means we use it under integrated pest management programs. Means there are certain ecologically sensitized areas where we have used too much of chemicals and we have damaged the land to an extent. Now in that areas we are not supposed to use any chemical which will further damage and it will convert into a bare land. Now under certain circumstances we can go with this one. It's called integrated pest management program. The pest is controlled by which pest you have to control, means then we'll go with the baculovirus. Right children, I think you understood. The next topic 10.5 microorganisms under biocontrol agents. Under that we discussed about microorganisms. Bt we discussed under bacteria. For fungi we discussed Trichoderma. For virus we discussed baculovirus. Baculovirus is species specific. Trichoderma will control the root-borne pathogens. Bacteria will kill off the caterpillar. Praying mantis is a predator for many insects. Ladybird is a predator for aphids. Dragonfly is a predator for mosquitoes. Now when we use chemicals, it will cause harm to our health. It will cause harm to the environment. That is why organic farmer finds to introduce the predator to kill the pest. Organic farmer's belief is biodiversity will future the health. He will tell that the more the variety, the more the sustainability, right? Now he will tell organic farmer will create a system not to kill completely, but he wants to check it, balance them at a level. On other side, if you go with chemical fertilizers, indiscriminately it kills the useful also, harmful organisms also, which is not good. Take a screenshot. We'll discuss with the last topic, microorganisms as biofertilizers. Right? So our last topic is microorganisms as biofertilizers in this chapter. Now fertilizers means urea is a fertilizer. Now again it comes the same thing. When you are using fertilizers, chemicals, that means you are damaging the soil. and it is expensive also. It causes environmental pollution and it's not good for health also, human health also. Instead, if we go with biofertilizers, now biofertilizers means living organisms which will enrich the quality of the soil, then they are called as biofertilizers. Microbes 10.6, microbes as biofertilizers. Microbes as biofertilizers. Now with our present day lifestyle, what we are doing is we are causing too much of environmental pollution. Now nowadays environmental pollution became a major cause of concern, right? Because of our present day lifestyles, we are damaging the environment, and now environment pollution, it became a major cause of concern, right? Now by using chemicals we have damaged the soil, water, land. But why did we do that? Knowingly, why did we do that? Because population is increasing. To the increasing population we have to give more agricultural products. If you have to meet that value, means you have to use fertilizers. For that reason you have done it. Now we understood the problem what we face when we overuse chemicals, fertilizers and all. So because of overusage of fertilizers and chemicals, what happened? The environment got damaged. Now there is too much of pressure on the farmers to switch off from chemical farming to organic farming. So there is pressure on farmers. What is the pressure? To switch from, they're asking them to switch from chemical farming to, organic farming. Now what do you mean by organic farming? Don't buy any chemical fertilizer. Instead go with biofertilizer, which is good for the soil. Now then, what are biofertilizers? Biofertilizers are microorganisms, right? They are organisms. They can also enrich the nutritional quality of the soil. So biofertilizers, what are they? Are organisms which enrich, means enhance, the nutritional quality of the soil. They will enhance the nutritional quality of the soil. Now what are the examples of biofertilizers? Under biofertilizers we can take the example of bacteria, we can take the example of fungi also, we can go with example of cyanobacteria also. The examples are, what are the examples, children? Bacteria, fungi and cyanobacteria. About bacteria we know rhizobium, right? A bacteria called rhizobium helps in nitrogen fixation in the leguminaceae plants. A bacteria called rhizobium, it will be there in the root nodules of the leguminaceae plants. It will be there in the root nodules of the leguminaceae plants, and what it will do, it will convert the atmospheric nitrogen into ammonia, nitrate and nitrite and gives it to the plant. So what is this association called? This is called symbiotic association. Rhizobium is doing nitrogen fixation by doing symbiotic association. Both are getting benefited. What is the benefit the plant is getting? Plant is getting nitrogen. What is the benefit the bacteria is getting? Bacteria is getting shelter and bacteria is getting food from the plant. So under symbiotic, rhizobium is an example. Under free living, if we have to take, the examples of free living bacteria are Azotobacter is one example. We can take the examples of Azotobacter and we can take the example of Azospirillum. Azotobacter and Azospirillum. Spirillum are the examples of free-living bacteria which can do nitrogen fixation. Rhizobium is an example of symbiotic bacteria which can do nitrogen fixation. Why to buy urea? Why can't we use rhizobium? Why can't we use azotobacter and azospirillum which can also do the same function? So this is a concept of biofertilizer. Now let us talk about fungi. We know fungi also will make associations with the roots. Those associations are called Mycorrhizal Associations. They are called Mycorrhizal Associations. What are Mycorrhizal Associations? Myco means fungi, rhiza means the roots of the plant. Now which fungi will make mycorrhizal associations? Means glomus. Which plant means Pinus, Cycas, Cedrus, all these plants. Now glomus will make mycorrhizal associations, symbiotic associations with the roots of the higher plant. Now what will the fungi do? The fungi called glomus will absorb more phosphate from the soil and it gives to the plant. They absorb more water also from the soil. They absorb more water and gives it to the plant. So what are the benefits the fungus is doing for the plant? Tell me. The fungus is absorbing phosphate from the soil, giving it to the plant. The fungus is also absorbing water and giving it to the plant. What the plant will do? Plant will give shelter, plant will give food to the fungi. Not only that, this glomus, what are the additional advantages? The glomus secretes growth hormones. The glomus secretes growth hormones and help in plant growth. It promotes plant growth, and this glomus which is there in the roots of the plant, it will not allow any root-borne pathogen to enter. So it protects the plant from other root-borne pathogens. It protects the plant from other root-borne pathogens. Not only that, such type of plants are tolerant to excess salinity and drought also. Plants have tolerance to salinity and drought conditions also. How many advantages the plant, the bacteria, the fungi is giving to the plant? See, first it absorbs phosphorus, gives it to the plant. Second, it absorbs water, gives it to the plant. Third, it produces growth hormones and promote the growth of the plant. Fourth, it protects the plant from other root-borne pathogens also. Next, it makes the plant to tolerate salinity conditions. It makes the plant to tolerate drought conditions also. Done with the fungi also. Now the last topic is cyanobacteria. Now cyanobacteria will be in aquatic bodies also. They will be in terrestrial bodies also. They can also do nitrogen fixation. Examples are nostoc, Anabina, Oscillatoria. What are the examples of cyanobacteria? They come under prokaryotic, right? So examples are nostoc, Anabina and Oscillatoria. So they do nitrogen fixation in the aquatic forms also, in the aquatic bodies also, and in the terrestrial forms also they do nitrogen fixation. Now this cyanobacteria are especially beneficial in paddy fields. Cyanobacteria are especially beneficial in paddy fields. In paddy fields, the cyanobacteria helps in nitrogen fixation. Not only that, when this blue-green algae, cyanobacteria, they die, they add the organic matter to the soil and make the soil fertile. That is one more advantage. Now, giving this awareness to the farmers, we have tried to convert the farmers from chemical farming to organic farming, making them to understand the advantages. Now they have switched, and now in India majority of the farmers are doing this only, and we have many biofertilizers available in the market. So children, in one shot we try to complete the whole chapter. If you like the content, like, share, subscribe to my channel. Thank you.